So in the previous section, we saw how decision making is related to the scientific process. And remember, the idea is that you need to get information in order to be able to estimate the probability that this condition or that condition is true. Whether it's looking at a correlation, how probable is it that the two are correlated, or looking at the distribution of a certain variable, how likely is it that this group of individual is taller than that group of individual. And so you need to gather this data, and this is what we're looking at here. And so one thing that's sort of key when you're starting to get data on a specific topic is to define your population. And that's important because you will want to sample that population in a way that is smart, in a way that is non-biased, and that gives you a good representation of the population. So, for example, let's continue with the example of height in humans. Let's say you think you want to characterize the height of humans in general, in this era. Okay, so basically your target population is the whole world. Okay, it might be different if you target people from a certain country or people that have certain conditions, right? And so you want to define your population very well so that when you consider a sampling strategy, how you're going to measure what individuals you're going to measure, you try to explicitly think about being uniform or having some way to avoid any biases. I give you an example. If you're trying to figure out the average height of people around the world, you might not want to stand outside a basketball stadium where all the tall players are coming out and have your only measurement made on these maybe not average individuals. And so you want some kind of random and broad sampling strategy that will allow you to be able to do a good estimate of the whole population. So here, the whole population, you select cities a little bit everywhere, and you will figure out some kind of way in these cities to get a sample that avoids all the biases. Okay, here I have another example. So to help you understand how you define your population, it's not always as easy as this case. Here you're trying to figure out, so let's take the example of bees. You have different species of bees, and they pollinate different flowers, and you're trying to see if there's a correlation between the species of bees in a certain patch or in a certain environment and how efficiently the flowers are pollinated. So you could ask yourself, what's the population here I want to sample? Is it the bees, the different species of bees? Is it the flowers? Or is it, in this case, I think it would be the different environments. So a certain environment has different combination of species of bees, and the flowers will be more or less pollinated. So you define your population based on that. Are you going to look at all different environments, fields, forests, or are you going to focus on certain type of environments, what countries? And so defining your population is important. Okay, so the idea is that you have a whole population here. You take your samples. You want to be able to get representative samples, in that you try to sample, as I said, without biases, and sort of in a way that will get at the whole variability of the population. And one good way of trying to sample something without biases is using a random process. So that's something that you will see often. It's important to think about your sampling strategy to avoid biases, and a good way is randomly picking which sample you're gonna, or which individual you're gonna sample. So here I have an example of people picking in a hat their shooting order or whatever. And we often do that in the lab in some way or another, whether it's picking numbers in a hat or throwing a dice or getting the computer to generate a certain sequence or um, certain order of the sampling. It allows us to avoid biases and avoid effects that we might not be able to think of. I give you an example. If you always sample your testing mood in teenagers, okay, and you're always testing high schoolers in the morning, and you're always testing middle schoolers in the afternoon. Well, you might have an implicit bias here. Maybe in average, the middle schoolers are sad in the afternoon, whereas for high schoolers, the morning is their best time and they're usually most happy, or vice versa, you know. The point is that you're introducing a bias here, and so your results will be skewed. So some kind of random process will allow you to avoid a number of biases. So let's take another example. And this time, what I'd like to look at is basically the type of variables you're getting. So here you have several patients. You're recording blood type, blood pressure, and weight. Okay. And so here, notice that first you have your sample size here, right? So you have 10 patients, that's your sample size. And for each patient, you measure 3 different variables. Now, this first column here, you could consider it a variable. It's the patient number or identity, or the question is whether this variable is going to be useful in making some kind of statistical analysis of your data set. And so the answer here is that to make some kind of statistical analysis, you need repeated measure, right? And so you would need to have several measurements of patient 1 and several measurements of patient 2 to be able to compare statistically if patient 1 and patient 2 have a different blood pressure, right? Otherwise, you can only say, well, at this time, what I recorded was different. Maybe in 10 minutes, it's going to be the same, or it's going to be the opposite. And so to evaluate the role of a certain variable, you need to have repeated measure. So sampling several times your target population or your target variable. So here, a patient is entered as a variable, but it's not going to be used so much in the analysis. We have 3 other variables here. And so we could, for example, look at blood type, compare blood type A versus blood type B. Do they have an average different blood pressure? Or we could correlate the blood pressure with the weight. Do people that have higher weight have lower blood pressure, or vice versa? And you could do those comparisons. Now, to start doing some kind of analysis on this data set, to choose an analysis, what's going to be very important is understanding the type of data you're getting. And here we're going to separate mostly this broad category here, quantitative data, or typically you use continuous data, but the quantitative data versus the qualitative data. And often it comes in the form of nominal data. And so quantitative is something you can measure, and typically that varies across a broad continuum. So it's measuring your height, it's the number of eggs that a bird might lay. It might be any kind of measurements that will vary broadly, and that can take many different values. Okay, so that's quantitative, and it's different from qualitative, where qualitative you can describe it, you can assign it categories, but it doesn't vary. It cannot take any value beside those defined sort of categories. And so the main one we'll be using is the nominal kind of variables, and nominal in name. So think nominal name, basically it's a category. Is it male or female? Is it this species versus that species? Is it treatment A versus treatment B? Okay. We have two other kinds of quantitative and qualitative that we're not going to use too much here, or we're going to assume it's the same as the nominal. So here the order, the ordinal variables, the individual arrive first, second, third in the race, for example. Is it the first offspring, second offspring? So some kind of rank. We're not using that very much for now. And the discrete, it's basically a special case of continuous, because you can imagine that if you measure height, well, what's the resolution you're going to use? Are you going to use, millimeters or fraction of inches? Or are you using whole inches, or is it whole like miles or kilometers? And so it might end up being a sort of gray continuum between the continuous and this discrete. Obviously eggs, or let's say you count the fingers on somebody's hands, the number of digits, that's pretty clear that it's discrete. But then at one point you might find variables you're not sure if it's discrete or continuous. Often it will not matter that much. And so we typically will talk about continuous variables. Okay, that's important because the kind of analysis you use really depends on the variables you get. And you'll see at first we will focus on analysis that has one nominal variable and one continuous variable. And especially at first we will use one nominal variable that's the variable we manipulate. So that's another aspect that I want to describe here. It's a certain perspective of how your experiments or your data set was collected, and what variable you manipulated versus the variable you recorded as the result of the experiment. So if you have treatment A versus treatment B and cancer rate, well, you have one variable that's independent. It's not dependent on the condition you have in your experiments, because you set those conditions. You give treatment A and treatment B, or you choose a cohort of male mice or female mice. And so it's independent, okay? The dependent one is what depends on your treatment, on your experiment, the result. So you measure cancer rate. It's not dictated in your experimental design. Now we'll see that at some points, the concept of dependent and independent might be a little bit blurry, and you record several things, but one might be the source of an effect you record in the other variable. And so you might have a gray area between whether a variable is dependent or independent, and that's often not very important. When it's clear, you can think of it this way to be able to choose the right test and to be able to present your result with a certain perspective. And in other cases, you won't be able to really describe it in these terms. And then you can choose all your variables as being dependent. That will be okay. I have another example here. So here it's whether the temperature in a room affects the scores of students taking exams in those rooms. So you might set the temperature to low versus high. So here you have a nominal variable, two categories that you set as independent variables, and you record test scores. And so test scores might vary between 0% and 100%, and this will consider continuous. Now this is an example where if you were recording ABC, well, would it be discrete, right? Or would it even be nominal? So you could record your test score different ways, and it could affect the exact kind of analysis you can use. If it's possible to use continuous, you have powerful tools that allow you to use one or several nominal variables as independent variables, and one or several continuous variables as dependent variables. And that will be the focus of the beginning of our class. Okay, so to summarize this section, we have a few concepts here that you'll find also in your text that you should remember and you should be able to explain and understand. And next, we will talk about experimental design. Thank you.